So now we're going to look at receptor tyrosine kinases, and the main function of these is the regulation of many fundamental cellular processes such as cell proliferation and differentiation. The structure is an extracellular ligand that has a binding domain. It's a hydrophobic transmembrane domain and an intracellular tyrosine kinase. So the tyrosine kinase we see right here, and typically it's known as an RTK. So in the inactive state, it's unbound to the ligand. So the two tyrosine kinases, or the receptors, haven't dimerized with each other to bind. But then what happens in the active state is a ligand comes in and basically joins the two receptor tyrosine kinase two RTKs. So now we have it in the active state. And so in the active state, what happens is the tyrosine kinase is already it has a phosphorylating ability because it is a kinase. So it's going to phosphorylate. And what will happen is it will call other compounds or other small proteins along to serve a job. So the first thing that's going to happen is it's going to call GRB2. So GRB2 will be called, and along with it GRB will have its associated compound, which is SOS. And SOS is a JEF. So if you remember, JEFs are used for phosphorylation. So it's going to convert a GDP into a GTP. And once this comes, RAS will come in, and RAS will now be in the active state where it now has its GTP bound instead of GDP bound. And then that will bring in RAF. And now what's happening is we're going down the MAPK pathway, which MAPK is kind of like this redundant pathway where it starts out as MAPKK, and then it has like a third K, and then it keeps losing a kinase and a kinase and a kinase to result in MAPK, and this is going to ultimately elicit a response. The second option is that we can have PI3K, which is going to get phosphorylated, and this is going to lead us to making PIP3. And if you remember, PIP is involved in lipid signaling, which is going to activate RTK. And this is going to originate as PIP2. The second is it could become PLC gamma that gets involved, and PIP2 is going to interact with PLC gamma, which is going to form DAG and IP3. And we've learned in previous videos that DAG and IP3 are involved with calcium release and cell signaling and cell proliferation. PKC specifically is involved in cell proliferation. So that's basically the gist of RTK. If you like the video, give it a thumbs up. If you have any questions, please leave it in the caption below. Be sure to subscribe.